So this one here was started in 20. And here we are going to look at the endomembrane system, and we have to know what that means, right? And also we're going to look at sorting and vesicular transport. So these concepts, at first, when you look at it superficially, it's not very intuitive. So you have to have a good gut feeling of what's going on here in order to understand what is happening with the process and with the molecules that are involved. You need to keep in the back of your mind what is the use for each of those molecules that is involved in these systems. Okay. So here are some of the objectives that we're going to cover. So basically, we are really going to focus a lot on nuclear transport. So we're going to revisit this role of GTPases and GAFs. Okay. We're going to look at nuclear localization signals. Actually, we're going to look at zip code signals, and those are really, really cool. Okay. And then we're going to look at transport into mitochondria and also into other membrane systems. Okay. All right. So when you're looking here at the endomembrane system, oh, actually, I should have updated this slide. Here, this title should actually say transport of things, okay? Transport inside of the cell. Because the endomembrane system is a system of membranes in which the inside part that fills up this membrane enclosed stuff or organelles, this inside is topologically equivalent, those are big words. Right? It means it has more or less the same environment as outside of the cell. So topologically equivalent means that it's more or less the same kind of stuff. Okay? So if you look here at the inside of the double membrane of the nucleus, you will see that it is continuous with the membrane. The outer membrane of the nucleus is continuous with the membrane for your endoplasmic reticulum. So the environment inside the endoplasmic reticulum is continuous with the environment of the inside of this double membrane of the nucleus. Okay, so that is the topological equivalent thing. And here is not showing, but after the endoplasmic reticulum, you're going to have a system here that is the Golgi. So the inside of the Golgi, because the things are budding out of your endoplasmic reticulum and going to the Golgi, so the inside of the Golgi is also equivalent to the inside of your ER. And then the Golgi, some of it is going to fuse to the cell membrane. So then, of course, it's going to mix with the outside stuff. So you can assume that the inside of those vesicles is also equivalent to the outside. And here is actually when we have this idea of transport. So this is exactly like that. Okay, so we're going to say here transport. Transport of stuff. Okay. So in order to transport things in the cell, especially when you're making proteins that are destined to certain membrane enclosed spaces, right? You're going to have to tell the protein where to go, okay? So let's see overall what is going to happen here. First thing that happens here, you're going to have your messenger RNA that is transcribed in the nucleus. Then you have the processing of that messenger RNA, and then you're going to have export of that messenger RNA to the cytoplasm. And now you're going to have meeting of mRNA with the ribosomes, right? So you have large subunit, small subunit, and of course we do not go into detail of translation, right? But there are a couple of things that you should know because you should have already taken classes that have taught you about translation. Yes. Who has no idea about translation in this class? Yeah, I know. Now you're like, I don't know if I put my hand up or not. Okay, I will put supplemental video that covers basics of translation, just in case you don't know what it is. But since you guys are really smart and almost graduating, I'm assuming that you have seen translation before. Okay? So a few things that you should know about translation is that your 5' cap helps your messenger RNA to find the ribosomes, right? And you have like translation factors that are important for the translation to happen. The translation of message into protein happens at the large subunit of the ribosome, and the ribosomal RNA is the thing that has catalytic activity. So the ribosomal RNA is working, or it is functioning as a catalyst. So it thinks it's a protein. Okay, it's an enzyme. It's working as an enzyme. It has enzymatic process. Okay. The peptide bond is formed in the large ribosomal subunit. And then to move those ribosomes codon by codon, you use energy, but this is one unusual thing about translation, that you use energy of GTP, not ATP, okay? So GTP is equivalent to the energy of ATP, but for the translation process, you use energy of GTP, okay? So that's the overall 5 seconds review on translation. Okay. So here, your RNA comes to the cytoplasm, you start to make your protein. Now, what is the side of the protein that you start to make? And now you have to remember, oh, what is she talking about? Primary structure of your protein is a sequence of amino acids. It's like, what? That was in previous life. We have to know that? Yeah. It's a good review for your cumulative final, right? All right, so what is the first part of the protein that is made? Is it your N-terminus or is it your C-terminus? Okay, the question is, when you start to make your protein at the ribosomes, you start to make those peptide bonds, what is the first part that comes out of the protein? N-terminus. N-terminus, right? So you start to make a protein, the N-terminus start to come out. So it makes sense to put the zip code at the N-terminus. Because if you want to send this protein somewhere, you know, you should direct it right at the beginning of the protein formation. Okay? So your zip codes are going to be embedded, or are going to be really coded at the N-terminus of your protein. So based on whatever the zip code says, well, it's basically the same thing as a US mail system. Right? So you look at your address and everybody has a zip code. So if you want to send a letter to somebody in Hollywood, Fort Lauderdale, you can put a zip code that is 33021, 33020, right? So you know your letter is going to be shifted to that side of town. If you want to send a letter to somebody in Coral Springs, you can put a zip code 33067. It's a different zip code. Your letter is going to be sent to another place in Florida. Right? So it's the same idea of zip codes. So when you start to make a protein, you're going to get the zip code, and the zip code is going to tell this protein to associate with target proteins that are on those membrane-enclosed organelles. So how is it that your protein is going to know where to go? Well, if you have a certain zip code that interacts with a protein that is on the mitochondria, then your protein is going to go and interact with that thing. And so on and so forth for the other membrane-enclosed organelles. Okay? We're going to look more into that, of course. So here, looking at membrane-enclosed organelles, what are they? Well, we have the nucleus, we have the ER and Golgi. We also have lysosomes and peroxisomes, mitochondria and chloroplasts. Those are all membrane-enclosed organelle. Okay. So here, there are 2 organelles with double membranes, actually 3 if you consider the chloroplast, but I tend to stick to animal cells. Right? So there are 2 membrane-enclosed organelles in which you find double membranes. Or okay, 3, right? 3, let me correct that. So 3. So you have here your nucleus, you have your mitochondrion, and you also have the chloroplast. Okay? All right. So here, when you start talking about this vesicular transport system, okay? So now we know that this is what I just mentioned about topologically equivalent environments, right? So now we know that if you have places that are topologically equivalent to each other, they are going to be more or less in continuity. So here, when you have the inside of the nucleus, you go here to the inside of the ER, and then also you're going to go and you see a lot of communication and transport from ER to Golgi. So those are topologically equivalent. Now, if you go and look at other membrane enclosed organelles like mitochondria or lysosomes, you see that, or even peroxisomes, you see that the inside is not equivalent to the other stuff that is in this endomembrane system. Okay, so those other organelles have distinct stuff inside of them. Okay, and you say, like, what does that mean? So let's say if you look inside of a lysosome, what you're going to find inside of a lysosome? You're going to find enzymes. Yes, enzymes. They're going to degrade stuff, right? And usually you have this more acidic environment. So if you compare that environment to something that is inside of the ER or the Golgi, you see they're totally different. Okay, and then let's say another example. If you look inside the mitochondria. Now you have two other subcompartments that are very much different from the environment that you find in the on the endomembrane system. So you call those organelles, you say that they are distinct or topologically distinct from your endomembrane system. So here is this idea of protein sorting. So here putting into a picture what I have just summarized very quickly about translation, right? So here you have your messenger RNA that is right there that found the ribosomes, right? So you found your ribosomes and you're going to start to make your protein. So here is your nascent protein coming out of the large subunit of your ribosome, okay? So this nascent protein, if your messenger RNA did not put a zip code in this protein at the N-terminus, then this protein is not going to go to a membrane organelle. That means the protein is going to hang around in the cytoplasm. So if the thing doesn't have a zip code, it will stay in the cytoplasm. Okay? Now, if this thing has a zip code, now something else is going to happen. Okay, is going to be directed towards some membrane enclosed organelle, or is going to be directed towards something that has to be excreted from the cell. Okay, so where is it that you put that little red zip code? Okay, it goes on the N-terminus, so that's right at the beginning of the synthesis of your protein, right at the beginning of the translation. Okay, how big is it? Well, it's not too big. It is anywhere between 15 and 16 amino acids long. Okay, so residues here really is for amino acids. So 15 to 60 amino acids long, so it's quite small. Okay. So now you are going to look at how that is going to happen. Okay. So looking here at the overall process of making a protein. Question here is what's going to happen to the protein? So answer here is that one of three things can happen to the protein. The first thing is it can hang around in the cytosol. Okay. The second thing is that it is going to be modified for export into specialized organelles. Okay. So if the protein is finished being made, it can be modified post-translationally, and then it's going to be imported into some organelle, okay, for function there, okay. And the third thing that is going to happen is that you start to make the protein, but because of the zip code, this protein is intended to finish translation in the rough ER. Okay, so now this protein is going to be transported to your rough ER for finishing up the translation. Okay, so that's the third thing that can happen to the protein. It can be shifted to the ER for co-translational import. That means it's going to be imported into the rough ER while it is finishing the translation process. Okay. Now, if you look at this thing and you say, this is confusing, now this is the part when you start reasoning things out, just thinking about it. Okay. You think, I'm going to translate protein. I need ribosomes. That's for all the proteins. If I start translating the protein, I have to start thinking, where is it going to go? Is it going to stay in the cytoplasm? Is it going to be imported to some protein? Or do I have to secrete it out of the cell? If I'm going to secrete it out of the cell, then it needs to be shifted to the rough ER. Okay? All right. So now, the first thing that I mentioned, it could just hang around the cytoplasm, and yeah, boring life, that's it, nothing else you have to know about that, except what the protein is used for. So here, the second thing that's going to happen, right? When this protein is meant to go, for example, to the mitochondria, now you have to shift it. You have to translocate it into this membrane-enclosed organelle. The protein is a big blob. You cannot just have a channel to allow this protein to go inside as a blob, because a big channel like that would allow a lot of other things to go inside of the cell, inside of the organelle, and that would be disastrous. Okay. So the thing you have to do, the thing you have to keep in mind is that first thing you need to do is to stretch out this protein. So you're going to need specific chaperones to stretch out this folded protein. And how is it that the protein gets folded in the first place? Huh? Naturally, right? Is an endogenous property of your primary amino acid sequence. It folds naturally. Now, to unfold it, it's going to be not very naturally. You need to put some effort into unfolding this protein. If you need to put effort into something, you're going to need energy. Okay? So those chaperones are going to use energy. All right, so you're going to have to stretch out the protein. You're going to have to bring it to some translocator, and then you're going to push it through the translocator to this destination, okay, to the protein destination. Once the thing gets inside of the membrane-enclosed organelle, then it's going to be refolded, which means you're going to have other chaperones, or it could just refold naturally. Okay, because the chaperones that are folded out here, they are not going to go inside the organelle. So they're going to be on the outside. So protein gets inside, it will fold again naturally. So we have details on that. All right. So here is third destination of proteins, right? So if you started making the protein and you have the signal peptide that is telling it to go to the ER, so this is what's going to happen. Okay? You have that little peptide that is going to interact with a recognition particle. Okay? So what is going to happen here? You're going to have here a signal recognition particle. Where is the signal recognition particle? It should be around here. Do I have a picture for that? Yeah, it should be right there. Okay. So this signal recognition particle, which is not labeled but is right there, is going to direct this signal peptide into another protein that is a receptor for the signal recognition particle. Okay? So while I'm talking about this, I'm giving you the names of these things. So first thing that recognizes the signal, if you have to name it, is recognize the signal recognition protein. Then you're going to bring it to a receptor, so you call that SRP, receptor. Okay? Once you bind to the receptor, you're going to have to shift the rest of this protein translation to go inside of the ER. There are several ways of doing that, but we're going to keep it simple. Okay? So just the main idea here. So once this thing is recognized, it is sent to the recognition to the receptor, and you are going to continue here the synthesis of the protein that is going to go through this channel that is called the translocation channel. Okay? Everybody cool so far? Yeah. Is this one right here, the blue one right here. This here is a ribosome. Yeah, it's this green thing right there. Yeah, you see this thing here that is interacting with the receptor right there, the green thing? That is the recognition. That is the signal recognition particle, SRP. Okay, so let me label this over here. This thing here, signal recognition particle. And it makes sense that it's going to interact right at the beginning of protein synthesis, right? Because it is meant to do that. Okay, so it recognizes it and it will stop the translation, and it's going to bring this whole thing close to this translocation complex. And the way it brings it is by interacting with the receptor for it. Okay, so this blue thing here is your receptor. This is your receptor right there. Okay. Everybody cool? So here, once you have your protein translocated, then you are going to get rid of your signal sequence. Okay, so let's see more details how that's going to happen. Okay, so here you're going to have your protein that is going to be translocated. It's not showing the whole thing. The protein is already there, folded inside. Okay, and close to this translocator channel, at the inside of that organelle, you are going to have one enzyme that is going to recognize the end of the signal sequence and is going to chop it off. But this is after your protein already got translocated to the destination organelle. Okay? Now, if you have to name that enzyme, what should you name it? Being the function of the enzyme should be giving it away. Signal peptide cuttase. Well, doesn't sound very scientific. So we call it signal peptidase, okay, which is right there. This enzyme associated to the inside of the translocator is your signal peptidase. It's going to cut the zip code, and then your protein is going to be folded inside of the destination organelle. Okay, everybody's cool with that? So main process here, you start making a protein, you have a zip code, interact with the sequence, with the recognition sequence protein. This thing brings it to the receptor, pushes the translation inside of the translocator, and at the end you cut the signal peptide off. This happens for proteins that are destined to be exported or to places that are membrane bound. All right, so everybody cool? There's a lot of information here. Okay. Yeah. Okay, the zip code, let me draw it here, is a piece of the protein right there that is made up of amino acids. So you have something like, glutamine, asparagine, threonine, tryptophan, a sequence of amino acids that is small, like 15 to 60 amino acids long. And that is it will fold in a certain way that will interact with a protein that is meant to recognize that stretch of amino acids. So the zip code is a stretch of amino acids. Okay, it's a small stretch of amino acids at the N-terminus of your protein, because it's the first one that is being made after you get to the ribosomes. Okay, so it's a structure of amino acids. That's all that it is, and the protein that recognizes it is just meant for it. Okay, you're welcome. Now, okay, so another information here: when your recognition protein interacts with the receptor, it is going to use energy, as I mentioned, but here again, it's not going to be ATP. It's going to be GTP energy. Okay. Remember that during translation, your ribosome is also using GTP energy. Okay. GTP, not ATP. Okay, so GTP energy is used for bringing this thing here together. Chat, GTP. I don't know about the chat, but yeah, but it's GTP. Okay. Yeah, you know what GTP looks like? It's a nucleotide. Yeah, and with a 3-phosphate, that's TP, 3-phosphate. Chat guanosine 3-phosphate. All right, so let's see what's happening here. So here is a more detailed picture of what is happening with this full translation import into your rough endoplasmic reticulum. Okay, so first thing that's going to happen, your recognition particle recognizes the zip code. This is in the cytosol. Okay, so this whole thing is going to interact. Okay, here is not showing the receptor, but remember you are using GTP for this binding, GTP energy right there, okay, for the binding. So now you are going to push your N-terminus into this translocator. And here a better detail of this is that this signal actually has a lot of hydrophobic residues. So hydrophobic amino acids will be pushed into an area of the protein that is hydrophobic, okay, and that is going to hold this signal at this translocator, while the rest of the protein keeps on being pushed through and delivered inside of that membrane enclosed vesicle. Okay. Now, after your protein is delivered over there, you're going to have to cut off this signal zip code. So this is where your signal peptidase gets into action. At the end, you're going to have your protein that is going to be at the destination. There are several things that can happen to this protein, right? It can go inside of this organelle. It can be stuck to the membrane. You don't have to know details of that. You just have to know the bigger picture of what is happening over here. Okay? All right. So here's just another picture showing the same thing. Okay. Let me see. Right. I just mentioned that. So here is your signal peptide. Here is your recognition particle. Okay. I'm not going to go into details of other proteins involved in here because I think this stuff is already complicated. Okay. So here the idea of things that are going across membranes. Okay. So once you have your protein translocated to the rough ER, which is right here, okay, rough ER, you are going to do post-translational modification of the protein, and you are going to start to make it ready for further translocation and deliver it to the destination where it has to go. So here is this bigger picture telling you what happens to the protein. Let's say you have a protein that is destined to be exocytosed, to be dumped outside of the cell. Okay? So this protein gets in there, it gets made, it's going to be folded, it's going to be modified. So this is going to be your smooth ER, okay, which is continuous with your rough ER. Okay? So now, out of your smooth ER, you're going to have vesicles, they're going to be formed, and they're going to transport this protein to the Golgi for further modification. The Golgi is this blob of stuff that is membrane enclosed, and it has several stages of Golgi processing. The part of the Golgi that is closest to the ER, you call this cis Golgi. Okay? The part of the Golgi that is further away from the ER and is closest to the cell membrane, you call this the trans Golgi. Okay? And then from trans Golgi, you can still make more vesicles and create the secretory vesicles that are going to dump the stuff outside of the cell. Okay? So the flow of stuff that is going to be secreted goes from rough ER to smooth ER to cis Golgi through trans Golgi and then vesicles and then exocytosis or destination. We have more details into that. So now we're going to start looking at the details on how that happens. Okay? So here is the first example of import of proteins. And this has to do with import of proteins that go to the mitochondria. Okay? So when you look at the mitochondria, remember that your mitochondrion, so let's draw mitochondria. Okay, it has two membranes, the membrane system, there you go, right? And then you have something inside of the matrix of the mitochondria that is circular and has a genetic code. What is that thing? Remember the circular DNA? Mitochondria has its own circular DNA. Yeah? What is DNA used for? Simple question. It's genetic information. That means it has genes that allows you to go through transcription and translation. So the mitochondria has genes to make protein. Now, why would you want to import a protein that is made in the cytoplasm of the cell? Why do you want to import it into the mitochondria if the mitochondria already has genetic code to make stuff? It kind of sounds redundant, right? But there's a lot of stuff we didn't talk about mitochondria. Mitochondria are actually very fascinating, right? Remember, it used to be some kind of bacterium, right? And then it got endosymbiosed. But it keeps its genetic material. But the problem now, or the issue now with the mitochondria, is that it doesn't have the genetic code for all of the proteins that it needs. It lost some of its genes, but it needs the proteins in order to function. The genes that the mitochondria lost actually got imported into the nucleus of the cell. So now the mitochondria, in order for it to work properly, it depends on the nucleus of the cell to make some proteins for it. So this genetic material that is inside of the mitochondria is not autosufficient. Okay? You are going to need, the mitochondria is going to need to import some proteins that were coded by the nucleus of the cell. This is why you have to go through this protein import into the mitochondria. Okay? Otherwise, the DNA, circular DNA of the mitochondria, would use its own ribosomes at the matrix here, and it would make its own protein. But it doesn't have everything, because the circular DNA is quite small. It doesn't fit everything that it needs. Okay? So here is what happens here. So here, you're going to have genes that are in the nucleus of the cell. Then, the proteins are going to made in the cytoplasm, not as finished product, but as precursor proteins. Okay? Now, those precursor proteins, they're going to have to be stretched out, and they're going to have to be pushed into the mitochondria. Okay? So a few things that are important here for zip coding of those mitochondrial proteins. The first thing that you see here at this zip code of of amino acids is that you're going to find a lot of positively charged amino acids. And now you have to remember, what are those positively charged amino acids? Right? Remember basic amino acids? You only have 2 of them to remember. You have arginine and you have lysine. So this zip code over here has a lot of arginines and lysines. Okay? Now, this folded protein is going to interact with proteins that are very good at unfolding stuff. So you call those chaperones. And in specific, you have a chaperone that is called HSP70. This is this pink chaperone right there. What this thing is going to do is going to interact with the protein and is going to stretch it out. In order to stretch it out, it needs to use energy, because naturally the protein wants to be folded in a certain way. So this chaperone is going to use ATP energy to stretch out that primary sequence of amino acids. Okay? So once you stretch it out, you are going to allow the interaction of your zip code with a recognition protein that is at the outer membrane of your mitochondrion. Okay? And it's obvious that it's at the outer membrane because this is the part that is in touch with the cytosol. Right? So now let's see what is going to happen here. This thing here is going to allow the interaction of your zip code to transporters. Okay? So transporters are channels, right? So here you're going to have a transporter that is in the outer membrane, and you're going to push this thing through. If your protein is intended to be at the intermembrane space, then you only have to worry with one transporter. And this transporter is located at the outer membrane. So the name we give to that transporter is a translocator in the outer membrane, or TOM. Okay, that is TOM. Right. So if a protein is going to stay in this space over here, you don't worry about the other translocator. Here is showing us one thing, but it's not. It's 2 things. This is one thing, this is another thing. Okay, they interact with each other. Okay. Now, if your protein has to go all the way to the matrix, now you have to worry about your inner membrane. Because if your protein only goes through TOM, then you're going to be stuck in this area here. To get into the inner membrane, you have to worry about this other translocator. And this other translocator, you call it translocator or transporter of the inner membrane, and that is TIM. Okay, so you got Tom and Tim. Okay, so if Tom and Tim are interacting here and your zip code is to go to the matrix, then you're going to go through this huge complex of pores. Okay. Everybody cool with that? Tom and Tim? Chaperones? So let's keep on going forward here. All right. So here, a bit more of a picture of what is happening. Okay. You are going to make this peptide that has your zip code. Zip code here is shaped as an arrow because it's going to point to where it has to go for a target. Okay. This is at the N-terminus. And it has a lot of positively charged residues. And then this thing is going to interact with chaperones to use energy for this thing to get imported. And here it gets to the outer part of the membrane. You find Tom over there. And then you find also the Tim at the inner membrane. So here is the picture of what is happening, right, between Tom and then Tim. Here it's showing that they are not interacting with each other, but you really have also the option in which they do interact with each other. Now, in this case here, it is showing what is going to happen in case your protein has to go to the inner membrane, but not get dumped into the matrix. Okay, so here are more details, painful details of what's going on here. So let's see what happens. We went through this chaperone business, we went through Tom, and now this thing is in the inner membrane space, intermembrane space, and is going to interact with a translocator. There are several translocators there. Okay, it's not only one TIM. You have a whole family of TIMs here. There are only two TIMs that you need to remember. Okay? One TIM is called TIM 22. This TIM is going to get the protein and is going to start pushing it through this membrane here, but is only going to interact with a zip code for proteins that are intended to be part of your inner membrane. So this is integral membrane protein. Why do you think mitochondria needs integral membrane proteins? Huh? Channel? No, integral membrane protein, in this case, is not going to be a channel. It could be. It is hydrophobic, yes. All right, now you guys are overthinking. We have that issue in the last exam that you're thinking so hard because this must be a very difficult question. And then I have people asking me like, oh my God, look at this question. It's like, just take a step back and make sense out of the question, because some of the questions are really easy, right? And now this is an easy question. Let's take a step back. Why is it you need mitochondria in the first place? When I say mitochondria, one sentence comes up to your mind. It should be powerhouse of the cell. Yeah, and that's so annoying. What does that mean? It makes ATP. It powers the cell. Right? Where do you make ATP? Inner mitochondrial membrane stuff, because you're going to have the electron transport chain. Remember that pain? It comes back. Electron transport chain is inside the inner membrane. And what is making up the chain? Those are integral proteins. And where do you think they come from? Right? Some of them get imported. And that's the job, to stick them in the membrane. That's the job of TIM, but not any TIM. That's TIM 22. Okay? Is it a catch 22? No, it's a TIM 22. And how are you going to remember that guy's job is to get things stuck in the membrane? Well, I always think about two little ducks that are swimming here in this inner membrane. Okay, this is a little eye and this a little big. Those are my two little ducks, happy ducks. TIM 22 puts stuff in the membrane. Okay? All right, so now let's see what else is going to happen. The other hand here, you have some proteins that have to go all the way into the matrix, right? So now if your protein has to go through this whole import and get into the matrix, you're not going to use TIM 22. You're going to use this other guy that is TIM 23. At least their side, sequential numbers. Right? So TIM 23. How are you going to remember that 23 goes to the matrix? Well, you're going to think about TIM 23. That looks like an N, sideways, 3. Does it help you remember? I hope so. Okay. So TIM 23 goes into the matrix. Okay? That's the transport of inner membrane. So here is again showing you what happens to proteins that are here going through the TIM and getting into the matrix. Now what happens to the protein once it goes through the matrix? Because remember, when it is stretched out and it's going to be pushed through these little channels, The chaperones that are in the cytoplasm, they are not going to go with this protein. Okay? So cytoplasm are going to be out there. Only the protein is going to be stretched and going to go through. Okay, it has to be skinny and go through this little pore. And inside of this matrix, the protein is going to refold. Most of the refolding happens by itself, but some of refolding needs help from other chaperones molecules. And it happens that it's about the same molecule that was out there, but this one is inside the matrix. Okay, so it's still HSP70, but this guy here is he lives in the matrix. Okay, so this is going to interact with the protein, and you are going to refold the protein inside of the matrix. Helps it to refold. Okay. All right. So now, do you have any questions about the mitochondrial import? You know the things you have to know about mitochondrial import? Nature of the signal peptide is positively charged. What happens when you stretch it out? Use chaperones, HSP70. Those use ATP energy, right? You go through TOM-TIM and you get into the mitochondria. If it's TIM22, it stays on membrane. TIM23, it goes inside the matrix. All right. So now we are looking at another type of transport, and this guy is your nuclear transport. So another example of a double membrane organelle. Okay. So now let's see here what the nuclear pore looks like, because most things that will go into the nucleus have to go through the nuclear pore complex. Okay. So if you look here from the outside, you're going to see that this pore complex has a lot of these fibers that are pointing out into the cytoplasm. Right? It has a central plug. And then when you look from the inside of the nucleus, you'll see that the pore looks like a basket, like from like a basketball hoop. Okay? It has this spoke-like rings and a central channel, kind of. Okay. So now we should ask the question, what is it that goes into the nucleus, right? Basically, anything that's going to work inside of the nucleus, like proteins that are going to help pre-mRNA, right? So we have also ribosomal proteins, and you have proteins that have a nuclear localization signal, okay? And all sorts of stuff that have to go into the nucleus, they're going to have to go through the pore. And on the other hand, things that come out of the nucleus, they also will have to go through the pore. Then what is it that's coming out of the nucleus? Well, tRNA, rRNA, mRNA, and stuff that has nuclear export signals. Okay? So here we're going to look at the nuclear import signals and then nuclear export signals. Okay. All right, so these are pictures I showed before. All right. So here is the deal of what is going on with this nuclear transport. Okay? So let's see here. When you are going to get some cargo imported into the nucleus, the cargo doesn't go by itself. It needs adapting proteins. Okay? Now let's see how that's happening. The cargo is going to interact with transportants. Transportants here, they can either be importing or exporting. In this case, this thing is an importing. Okay. And what is going to happen here, that transporting has to be at a specific conformation to recognize the signal sequence of that cargo. Okay, that's a zip code. So zip code for the cargo interacted with this transporting, and it is going to allow, once this thing binds, it's going to change the conformation of that transporting protein, and is going to expose binding sites that are going to interact with those proteins that are at the nuclear pore. So the proteins that are at the nuclear pore, you call them nucleoporids. Okay? So transportin interacts with nucleoporids and bring in the cargo. Okay? This guy gets in there, bringing the cargo. When this thing is inside of the nucleus, it is stuck to the transportin. So it's useless. You gotta get transportin out of there. Okay? So what do? In order to get transportin or this importin to stop interacting with the cargo, you're going to use another protein to go over there and bind the transportin. So this protein over here is inside of the nucleus, okay? Inside of the nucleus, and it has a site that can bind either to GDP or GTP. Okay. If this site is bound to GDP, it is not going to interact with transportin. If the site is bound to GTP, now it is capable of interacting with your transportin. Okay. So what is happening over here? Inside of the nucleus, you have to get GTP to interact with this other factor. So to get GTP in there, you're going to kick out GDP and you're going to put in GTP, and there is an enzyme for that. Remember the GAF? That is the GTP exchange factor, okay? And that is inside of the nucleus. So inside of the nucleus, you're going to have an abundance of GTP, more than what you have in the cytoplasm, right? And you're going to have a presence of this GAF. So GAF is going to get GTP and is going to plug it into this other transport protein. So GAF does the exchange, and now this guy is able to go over there and pluck out the interaction of that importin from the cargo. Okay? So now the cargo gets delivered, and now this association protein is stuck to this transportin. And if it stays in the nucleus, it is useless. So this whole thing gets exported back into the cytoplasm. Okay? In the cytoplasm, you know that this adapter protein is going to have GTPase action. Okay? So this thing is going to hydrolyze GTP, and this is what is shown over there. You're going to use water to hydrolyze GTP and get this thing to be GDP again. When this thing becomes GDP, now you are going to dissociate your transporter, which is your importin. I know it's a lot of information. This thing is very packed with information, and I hope I'm not giving you a headache. So I'll have pictures for that. Okay? Let's look into more details of what's going on here. Step by step. That's important stuff. And this is when I had to do this animation before AI. So it took me forever. Okay. If you ever did animation before AI, you know what I mean. So here, what's happening? Import of stuff. You have your cytoplasm, you have your nucleus. And here is your nuclear pore, a couple of them. You have your importin. That's not the cargo. Now you have a cargo associated with importin. Cargo got picked up, and this thing is going to be imported. Okay? It got imported. Inside of the nucleus, you want to have more energy. So you think that the inside of the nucleus, you're going to have, if it helps you to remember, nuclear energy is more energy than what you're going to find in the cytoplasm in the form of GTP. Okay? So inside of the nucleus, what is going to happen? You are going to have a couple of important players. Okay? You're going to have that adapter protein that we've seen before. That adapter protein, the name of it is RAN. And RAN interacts with GDP. RAN is the guy that has GTPase activity. Okay? But inside of the nucleus, okay, it is interacting here with the GDP, and it needs to have GTP in order to interact with your transporters. How does it get GTP? By action of this enzyme. That's your GTP exchange factor, GEF. Okay, so you put GTP on RAN. Now RAN can go over there and say, all right, let's dissociate this cargo from the transporter. So cargo gets dumped in the nucleus. This thing inside the nucleus is basically useless, so it needs to go back out in the cytoplasm. So now, okay, let's get this picture here. To get back out in the cytoplasm, when it's interacting with transporting in the GTP form, this whole thing has high affinity for the pore and is going to get exported. Okay, don't get into crazy details of exactly what is happening. Those are transportants. Okay. When you get to the cytoplasm, remember RAN has GTPase activity, is going to hydrolyze GTP. Once it does, you're going to dissociate the transportants. Does that look less complicated than everything in one kind of diagram? Hopefully. So that was the idea of nuclear import and nuclear export. Okay. Is that well understood? Do you have questions on that? No. Okay. Now, I am going to stop here. And yeah, we didn't get to iClicker, but I will compensate it for next class. Now, one thing that I want to mention, pay attention, yes, pay attention, because we